Okay, so today we're going to be talking about G protein couple receptors. So G protein couple receptors are 7 transmembrane receptors. This simply means that they will cross the membrane several times from where they are the receptor to the inside of the cell. And we're going to take a look at that a little bit more closely here in a little bit. They are only found in eukaryotes. So that's going to be human cells and animal cells. They're not found in plants or bacteria. Because they are so versatile and involved in signaling pathways related to many diseases, they are the targets for 30 to 50% of modern medicinal drugs. So they're very popular with the pharmaceutical industry just because they are very characteristic of themselves. So they're able to design medications and drugs for these types of receptors that will be applicable in many different areas. So conformational change is caused by a ligand. So when the ligand binds to the receptor, there is a change in the conformation of that receptor itself. This initiates intracellular signaling events that ranges from neurological to cardiovascular, endocrine, reproductive function, physiological processes. These are a whole bunch of different areas that it affects, and it's not limited to those. There are many different types of G coupled or G protein coupled receptors. They also range in size. So they're very small molecules that can bind to these receptors, but they can also bind to peptides like a polypeptide chain or a very large protein. And we're going to talk about some ways that they'll bind differently to the receptors themselves, depending on the type or size of ligand that we're talking about. So different processes and effects of the GPCRs are still being studied today. There's so many different things about these receptors. There was actually a Nobel Prize awarded in chemistry for studies in GPCRs as recently as 2012. So there's still a lot to be discovered about these specific receptors, their signaling pathways, and how to utilize these for treatment in humans, primarily for treatment in pharmaceutical industry is a primary cause and reason for all these studies that they're doing. So let's talk about the way the GPCRs work. Okay, so this slide shows the structure. We have that image there is not necessarily the typical image that you're going to see of this type of protein. And again, we're going to look at some other graphs and diagrams of that in a little bit here, but I like these ones because it's a little bit more of that typical protein structure that we would actually see if we were looking at the protein more on a microscopic level, but we're going to look at it in more of like a learning level that's a little easier to visualize. So let's see, the GPCRs are transmembrane. That means that they go all the way across the membrane. The protein that spans the cell membrane 7 times and contains intracellular and extracellular domains. So you can see on this image here, this is going to be our extracellular part. So that's outside of the cell. This is our cell membrane here, where we would have our heads, our tails, our heads again, and then now we're intracellular down on this bottom part. You'll notice most of the protein resides on that end here. And then this just second image here, you can see the ligand binding to the external receptor. That's on the extracellular side. Goes across the membrane. And then these are some subunits over here, the gamma, beta, gamma, and the delta there. So we'll talk the G protein portion. We'll talk about that here in a little bit here. So this is a tertiary structure. It is made up of connecting loops as well as terminal extension sequences. So the GPCRs bind ligands. So that's this little guy here, that yellow circle on the top there. Within their transmembrane and or extracellular domain. So the ligand binding elicits conformational change that initiates downstream intracellular signaling events through arrestins and G proteins. G protein couple receptors are one of the largest gene families in the human genome, with more than 1,000 different types of GCPRs. That's a lot of different proteins and receptors to cater to in the human body and to be able to potentially treat with pharmaceuticals. They encode about 800 different proteins. Because GPCRs possess a unique structure and are available for ligands at the cell surface, they are utilized by the pharmaceutical industry for therapeutic treatment. So the G protein, it can be single subunits, but in the case of GPCRs, all G proteins are heterotrimeric. So that means that they have 3 portions, trimeric, 3 different subunits. It's going to be the alpha, the beta, and the gamma subunits. And you can actually see in the beta and the gamma, so this kind of pink color and the yellow color, they have lipid anchors. So that's this little blip, the tail kind of thing that looks like it's coming up into the membrane there. That's a lipid anchor for those specific subunits themselves. So the GDP binds to the alpha subunit when the protein is inactive, and when the protein is active, it's going to be GTP. And we're going to come back around to that too. So let's take a look at our next slide here. So binding and conformational change. Ligands bind to extracellular N-terminus and the loops, so the glutamate receptors. Binding sites within transmembrane helices are the rhodopsin-like family. So if we look here at this image on the top right, you can see that not only are you able to bind at that N-terminus, but you can actually bind on these little loops that come across as well. So this is the first time we're seeing a good way to count these loops. So the 7 transmembrane is we're going to go down 1, 2, 3, 4, 5, 6, 7. That's where that name comes from. It goes all the way across 7 times. And then the end of it here is the C terminus, where you can see where it says that here. So they are all activated by agonists, although a spontaneous autoactivation of an empty receptor can also be observed sometimes. So I like this picture down here at the bottom, because this one on the left, where the image A is it shows the active versus the inactive state of the protein. So you can visually see how there's a conformational change between those 2 images. And again, this is the way it really looks as opposed to the drawings that we do, just so we can conceptualize that a little bit better. So the agonist bound beta to AR as it transitions from an active state to an inactive state, with this A is the non-hydrogen atoms represented as lines, and the B is the protein backbone represented as ribbons. Transmembrane helix 6, or TM6, is the colored red to highlight its high degree of mobility during the transition between active and inactive. G protein coupled receptors activation is accompanied by the outward movement of transmembrane helices 5 and 6, which create a cavity on the cytoplasmic side of the hepatohelical domain. Another point here, the agonist, I'm getting a little tongue-tied. The agonist activated GPCRs act as a guanylyl nucleotide exchange factors for heterotrimeric G protein. We'll explain kind of what that means a little bit more. In the receptor-bound G protein, its nucleotide binding pocket opens, which results in the loss of the GDP, occupying this site in the inactive form. And that makes a spot available for the GTP, which can then bind. It is much more abundant in the cytoplasm. So once that GDP disassociates, we can have the GTP come and join on that alpha subunit because, like I mentioned before, it's a lot more common in the cytoplasm. So we're going to actually do a little drawing here where we can visualize some of these changes within the cytoplasm itself. So I added this image to the PowerPoint so you can review that later if you'd like. But we are going to work through that together here. When my computer decides to cooperate. There it is. Okay. So this is our cellular membrane here. So as you can see, we have the hydrophobic heads. Get my pen out. So these are our hydrophobic heads here, these black circles on either side of the membrane. And then they have a those are hydrophilic heads, hydrophobic tails. So hydrophilic heads means that it likes water, right? So the inside of the cell, remember, is filled with mostly water. So we need the heads that like the water, the hydrophilic heads, to be on the cytoplasm side and also on the other side of the cell as well. But the hydrophobic tails is what keeps things from passing through. So that's where we need these receptors, and that has a lot of other plays with it, but just as an establishing and remembering the way our cell membrane works, this is how the original design is. So the extracellular portion is going to be on the outside here. Extracellular. And this is the intracellular. So on this intracellular part, this is where we have the cytoplasm. We're going to have, our nucleus, and all of our fun stuff is going to be on the inside of our cell here. So when we're drawing our G protein now. So remember, it's going to be a 7 trans membrane. So we got to cross the membrane 7 times. So we're going to come across and go, oop, that didn't work, did it? Let's try again. We'll go 1, 2, 3, 4, 5, 6, 7. And again, we looked at those kind of curly Q options before, but just so we can visualize, we're going to kind of spread it out and look at it this way. So I drew right up here, this little half circle up here. This is going to be our primary spot where the ligand will bind. But do recall that we can also have bindings on these loops up here as well. Different types of protein of ligands will prefer to bind on those loops. So we'll come down on like that. And now we're going to draw let's go with yellow, our alpha subunit. Remember, it has that little lipid tail up in here, and it's going to come around like that. Will be our alpha. And then let's do our gamma, that same structure. Tail up like that. Oh, this will be our gamma subunit, and our beta. Let's make this our beta subunit. He doesn't get that little tail, but what we're going to see here is that the beta and the gamma subunits are going to kind of travel together. They're going to be little buds there. So as we come along, this is right here with all these 3 together. This is our G protein. G protein. And we can see how it's associated with the 7 transmembrane receptor right there. So now let's take a look at that if it gets activated. So we're going to come across 7 times again. 1, 2, 3, 4, 5, 6, 7. Got a little spot for our receptor. This time, oh, you know what I forgot to put on here before was this is going to be GDP. And that's important guanine diphosphate because we have one less inorganic phosphate molecule here. So it's diphosphate, DP, as opposed to what's going to be TP, which is going to be our active form. So we'll draw our alpha subunit here. And now the alpha subunit has GTP. So it has an extra phosphate. So that makes it active. And we'll put our gamma and our beta, which like to hang out together. They have separated. And they're going to go off and cause their own reactions later. But for now, let's look at this. This is our initial is on the left. And once we have that conformational change here, we are able to separate these two units, which are going to have a lot of other associated factors. And recall that we do need that once that receptor is going to bind. So the ligand will bind right here to the receptor. Most often, this is going to be the kind that we're talking about. Do recall that we can't have them actually bind here, and they will just elicit other reactions. But that's what we're going to look at here. So let's pop back over to PowerPoint here. Okay. So that's just kind of that image there. See if I can get my iPad back together really fast. That we were looking at. So just to kind of regroup what we talked about there, where you can still see that same picture, the GDP disassociates from the GTP, when the GDP disassociates from the alpha subunit, the GTP is then able to bind in its place. Because remember, there's way more GTPs on this lower side of the screen, which is the intracellular portion of the image. And then the GTP binds. The alpha subunit disassociates from the beta gamma dimer. Now we're going to call it the beta gamma dimer because those two are stuck together. Before it was the 3, now it's just the 2. It used to be heterotrimeric. It moves away from that alpha subunit. The alpha subunit leaves and alters and regulates functions of a target protein. So now it's going to go on its merry way and do its job, which we'll look a little more into that here in just a little bit. The beta gamma dimer can regulate functions of other proteins. It's less common, but it can actually do its job as well. So with one ligand, we now have 2 different potential subunits going on and starting other reactions. So the freed active receptor can bind and activate another G protein molecule, which provides the signaling amplification at this level. Okay, so here's this out of the way. Here's some signaling amplification. So you can just look at the image there, which is the homodimerization, the intramolecular cooperation. So you can see how the signal is amplified just by multiple responses. The G proteins affect ion channels or enzymes. They stimulate or inhibit activity. And they do a lot of things, these G proteins. As long as the ligand is still attached, the action will continue. The activation of a single effector molecule will induce the movement of numerous ions across the plasma membrane or the conversion of many substrate molecules into products. This is how the signaling amplification is accomplished. So now let's work through another image here. So this is the alpha subunit here. These are just some general steps. It is bound to GTP instead of GDP. In this example, the alpha subunit regulates a membrane-bound enzyme. The enzyme then serves as a catalyst for a reaction producing a second messenger. The second messenger serves as an amplifier for the initial signal through the use of protein kinase activation. So protein kinase phosphorylates other proteins. Some are also enzymes. Once these proteins have been phosphorylated, their biological activity is altered and a response occurs. So let's look at CAMP on this next slide here. So this is a very busy slide. And we're going to kind of take it piece by piece here as we go, and it'll make a little bit more sense that way. Okay, so on this slide here, the alpha subunit is the S type. Then we are working with a GS protein. The activated subunit stimulates adenylyl cyclase enzyme. The ATP is the substrate used, and it will produce CAMP, which is the second messenger. CAMP, activated cycle AMP dependent protein kinase, which is the pKa, has two regulatory units, two catalytic subunits. Once bound to the regulatory units, the catalytic subunits separate from the regulatory units and become active. Now, the now phosphorylated is ready to bind to the promoter or the regulatory sequence controlling transcription of the target gene. Counteracting mechanisms include the protein phosphatase, dephosphorylated activated proteins, leaving them inactive. This counteractive measure is a way of controlling the signaling pathway, so it doesn't just keep going forever and ever. This is called negative feedback. So the pKa, the phosphorylated phosphodiesterase, which is the target protein, it actually hydrolyzes the CAMP into 5-AMP. This keeps the pKa brief and also localized. So this particular process plays a role in learning and also memory. Okay. So this is another couple of steps from that big picture that we looked at first. So this is going to be the GI and the GQ proteins. So the GI is you can see is on the left there. It's kind of the shorter series. Is the adenylyl cyclase enzyme decreases the CAMP. So because of that, the pKa is never activated. So it's the opposite of the GS. It's a lot shorter cycle. It doesn't have all of that extra CAMP attached to it. The GQ, however, is going to be a little bit more complicated here. So it stimulates phospholipase C to cleave the membrane lipid, which is the PIP2. This reaction leads to the products DAG, which is diacylglycerol glycerol, and it remains in the plasma membrane. IP3, which is inositol triphosphate, travels into the cytosol and binds to the IP3 gated channel in the endoplasmic reticulum. Now that recall that there's calcium is highly associated with the endoplasmic reticulum. So now that we're bound to that, calcium is released from the endoplasmic reticulum. Together with DAG, the calcium-dependent protein kinase C, PKC. PKC is catalyzed in the phosphorylation of cellular proteins. These mediate cellular responses. The second messengers in this process are going to be the IP3, the DAG, and the calcium. Okay. So now let's look at some of the ways that these signal pathways are actually stopped. So the ligand separates from the receptor. So when it's no longer attached to that little piece on the top, this is going to be the resting state. So the G protein utilizes GT apase, which is an enzyme, and it's going to actually hydrolyze that GTP to reform the GDP. So it takes off the inorganic phosphate, leaving us with GDP, which is the inactive state. We go from tri to di. The alpha subunit reassociates with the beta and the gamma subunits, and we reach our resting state. Another process for this is inactivation of the receptor. In this case, the GPCR activates the G protein and also the GPCR kinase, GRK. GRK and pKa and PKC phosphorylate the GPCR. The now phosphorylated GPCR will bind to a protein called arrestin. This prevents this prevents interaction with the G protein and the receptor. It also couples the receptor to the endocytosis machinery, inducing internalization of the GPCR. So let's look at a couple examples of this and help relate this to real life. So let's look at epinephrine. Epinephrine is the ligand that binds to the receptor. So imagine that, it's going to be too hard for me to pull that back up, but imagine that our little green dot is epinephrine and it binds to our receptor at the top of the squiggly 7-transmembrane protein. So it's the ligand. The GPCR functions as an adrenergic receptor in this situation, and it undergoes the conformational change. GDP is swapped for GTP. The alpha subunit finds the protein adenylyl cyclase, regulating its function and stimulation. So it breaks away and goes and finds its protein. The adenylyl cyclase converts adenosine triphosphate, which is ATP, to cyclic adenosine monophosphate, which is the CAMP. Loses 2 phosphates. CAMP is the second messenger, and this causes increased heart rate, dilated skeletal muscle vessels, the fight or flight response, and a breakdown of glycogen to glucose, because we're going to continue to need that energy. Another clinical correlation here is cholera. It comes from the Vibrio bacteria, and this releases toxins that alter the alpha subunit. And this disallows it to hydrolyze the GTP to GDP. So recall GTP is the on, so it's stuck on. Because it is stuck in an activated state, it will continue to activate adenylyl cyclase indefinitely. And this leads to CAMP will be persistently elevated. Clinically, patients will suffer from diarrhea because the chloride and water continue to flow into the intestinal system, resulting in severe diarrhea. So conclusion, a little wrap up of what we've talked about here. The G protein couple receptors are utilized throughout the body for many purposes. Although they have different ligands and different actions within the body, they share that 7-transmembrane alpha helices and alpha, beta, and gamma subunits. The alpha subunit is active when GTP is bound instead of GDP, leading to the separation of the subunits and further actions within the cell. G proteins regulate a wide variety of human physiological processes, making them a great target for pharmaceutical intervention. All right, thanks guys.